Cell communication. Cells communicate from cell to cell through direct contact or secreting regulators. Cell communication is then broken down into three parts: reception, transduction, and response. Reception is the detection of a signal molecule from an external source. Transduction is converting the signal so that it can bring a specific response on the cellular response. Response is a cell's response or reaction to the signal molecule. Now we're going to break down the three separate parts. A signal molecule, or a ligand, and a receptor bind together. This binding is highly specific, just as puzzle pieces fit directly together. Receptors are found in two separate places. First, there are intracellular receptors. These are found in the plasma membrane of the fluid mosaic model. They must be hydrophobic because it crosses over the membrane without help from a protein. The second type of receptor is a plasma membrane, which binds to water-soluble ligands to get it across the membrane. Here are some examples of a plasma membrane receptor. A G protein coupled receptor helps bind ligands to a G protein. This causes GTP to displace GDP and activates the G protein. Next, the G protein activates an enzyme, leading to a certain cellular response. Another example of a plasma membrane receptor is receptor tyrosine kinase. Signal molecules bind to the receptors. Together, they create a dimer, and each tyrosine adds a phosphate. This creates two cellular responses. Then we have a ligand-gated ion channel, which is found inside the membrane and opens and closes for ions to pass through. This regulates the ion flow. The ligand is used to open the gate and allow the ions to pass through. Transduction uses an enzyme to produce a phosphorylation cascade, which amplifies the signal like a megaphone, which activates proteins at the next level. A second messenger is a non-protein water-soluble molecule to make sure the message is received. Once activated, it can initiate a cascade. A response regulates protein synthesis by turning specific genes on and off in the cytoplasm. This regulates the activity of proteins, either by lessening or increasing their production. This is like a light switch, turning the genes on and off. To look at cellular communication as a whole, apoptosis is the perfect example. Apoptosis is a program of controlled cell death and suicide. Once the signal is received, the cell is dismantled and digested by itself. In summary, this flowchart is an example of how cell communication is broken down. Thanks for watching. Until next time.